Please off your mic. Please off your mics.
Yes, she can start now.
Dr. Deshmukh,
Yes,
today we have Dr. Anmol Randhawa from MIT University, Mohali. She'll give us a very good talk, and she's a very good speaker also. So Dr. Deshmukh, you can say a few words before we start.
Thank you, sir. Really, today I'm very happy that one of the madam from Amity and going to share her research, her work in the lignocellulose. And this lecture is being recorded permanently on the YouTube of Microbiologist Society. And it will be made available because we have one group only of our research students. We'll circulate this, and I'm sure many research students will enjoy this lecture. So thank you, sir, Swarnajit Singh, sir.
Thank you, sir.
For bringing such a eminent personalities in the subject throughout the India. Thank you.
Thank you, sir. Thank you very much. Thank you, Dr. Deshmukh. Now, Anmol, I welcome you for the talk.
Thank you, sir.
Before that, you please give a brief introduction about yourself right from MTech onwards.
Yeah, sure.
Yes.
Thank you, sir, for this opportunity. And so I'm PhD from CSIR Institute of Microbial Technology, Chandigarh, and I'm from 2008 batch. And my supervisor was Dr. Alok Mondal, who is now a professor at JNU, New Delhi. And my expertise lies in yeast molecular biology and biochemistry. And after my PhD, I joined ICER Mohali for an year for doing postdoc in Schizosaccharomyces pombe. And my topic was protein degradation pathways. And since 2015, I have been working with Dr. Shams Yashdani on for the development of lignocellulolytic enzyme technology for 2G biorefineries till 2022. And
please off your mics. Hello. Please close your mic.
And so today I'm going to share my research work that I have been doing last 8 years. And that is for the development of lignocellulolytic enzyme technology for second generation refineries. And my work is basically required synthetic and system biology. I request everyone to please mute your
Hello. Yes. Okay. Yes.
Okay. So my talk will be, it will have major 5 segments, 5 sections. I've divided my talk into 5 sections. So first I'll introduce the topic, what are second generation biorefineries and what is the role of lignocellulolytic enzyme technology in the second generation biorefineries. So then I'll talk about how do we tame the fungi that we isolate for doing this kind of work. And then how did we develop the molecular toolbox for genetic engineering of fungi? And then what are the ways how we rewire the signaling networks to increase the cellulase production? And my final segment will be the scale up and submerged fermentation to produce lignocellulolytic enzymes at 20-liter fermenter level. Okay, so first, let's start with biorefineries. So what are biorefineries? So they are biological systems that produce fuels and chemicals from biomass. So all kinds of these systems, we call them biorefineries. So biorefineries, they are classified on the basis of biomass that they use. So first generation biorefineries are those where we use food grade sugars, starch, vegetable oils, animal fats, and with the help of microorganisms, we convert these sugars, these energy rich resources into biofuels or biochemicals, biopharmaceuticals, or power or any other kind of biomaterial. The next level is second generation biorefineries, which use the renewable biomass resources, which are not food grade and are present in ample amounts in our country, and which we can convert it into biofuels, biochemicals, and other biomaterials with the help of microorganisms which can utilize these kind of bioresources. The third generation biorefineries are those which generally utilize the microorganisms which are photosynthetic in nature. So they are the most effective ones because all they require is sunlight and water and oxygen, and they produce biomass for us. And that biomass then can be utilized to produce all kinds of products for industrial and other uses. So I'll be talking about the second generation biorefineries. So let's first have a look at the basic structure of second generation biorefineries. So what are these? So all kinds of biomass which is recalcitrant, means which cannot be digested and cannot be utilized for human use, they are used as energy source for these kind of biorefineries. So since these biomass, they are made of lignin and cellulose. Both lignin and cellulose, they are very hard material, and and enzymes, they cannot break them until the structure can be broken. So first we need to pre-treat these biomass by either acid or alkali so that we can break the structure of these molecules so that enzymes can penetrate inside and break down these carbohydrates into fermentable sugars. So once pre-treatment is done and the basic skeleton of the biomass is broken, and then enzymatic hydrolysis happens with the help of enzymes called cellulases, where these cellulose and hemicellulose, which is converted into fermentable sugars. And then once this happens, these fermentable sugars, which is either 5-carbon, that is xylose, or 6-carbon, that could be glucose and other hexoses, they can then be converted into useful fuels or biochemicals with the help of microorganisms. So this is the basic structure of second generation biorefineries. Okay, so now what is the importance of fungal cellulases? So fungal cellulases, first of all, why they are important? Because fungi they are saprophytes, means they have the intrinsic ability to digest all these insoluble recalcitrant carbohydrates and utilize this energy for their survival. So they have the intrinsic ability, so that's why they are the most efficient ones, and they are the most gentle way to break all other methods are chemical based methods. This method gives us an advantage because it's a gentle method, it's economical method to convert this lignocellulosic mass into fermentable sugars. Another important advantage of fungal cellulases are that they are secreted in large amounts outside the cell. So we don't have to break the cell. So the extraction is very simple and it doesn't require a lot of efforts and energy and instruments. So enzymes are secreted out of the cell. Another important feature is they are very fast growing eukaryotes than most other eukaryotes. So we can have the product in few days time at a fermenter level. So when I joined the lab, bioprospecting was done by a PhD student in the lab, and he identified that there is one fungi, that is Penicillium funiculosum, whose secretome is actually all the enzymes which are secreted outside, and together we call it secretome. So he found that the secretome of Penicillium funiculosum, it has the better saccharification abilities, means it has the better hydrolyzing capacity. So the enzymes are much better than rest of the fungal isolates that he screened. And he also found that the secretome was even better than commercially available cellulases present in India, as well as it was equally found good with the world leaders that are novozymes. So the secretome was good. We had one organism in hand. Now the task was to make this, to make the enzymes at large scale and in the economical way. So the first task was to understand the physiology and growth of the fungus that was there and that we had found, that is Penicillium, and to understand how does it produce, what is the mechanism of cellulase production by this fungi. And we found that the glucose, whenever fermentable sugars are present, the fungus doesn't produce any kind of enzymes. Only when it faces some kind of carbon stress or when there is no other sugar, free sugar present in the environment, only then it produces cellulases. So if you will culture it in the glucose or any other free sugar, it will not produce any cellulases, but it will produce a lot of cellulases if you will feed cellulose or any other kind of recalcitrant carbohydrate to it. Then we also did growth and media optimization to see the best way to produce celluloses from this fungi. And the last was to, since our next step was to do the genetic manipulation to increase its capacity to produce a lot of cellulases. So we also wanted to see what is the what kind of antibiotic sensitivity is present for this fungi. So we checked majorly 3 antibiotics. So they are hygromycin, zeosin, and norceothricin. So these 3 antibiotics we found that all 3 antibiotics, the fungi was sensitive towards these 3 antibiotics for first 48 hours. But if we were leaving the culture for longer periods of time, the fungi was able to grow even at a concentration which is higher than minimal inhibitory concentration. So it can grow equally well after prolonged time. After 48 hours, it was able to grow. And we were not able to find out why it was growing even at the at such high concentrations of the drug. So our first task was to find out if the genome already has the genes for resistance towards these antibiotics, and we found none. The genome of Penicillium funiculosum didn't had any resistance gene present in them. Then we wanted to search what is the real reason why the fungi was able to grow in presence of these antibiotics. And for that, we looked at the genomics of the fungi, and we found that around 600 different kinds of drug transporters were present in the genome of the penicillium. And they were of two types, ABC transporters and multi-facilitator system transporters. So they make, as an ABC transporter, so they make 15% of the total transporters present in the genome, and MFS, they made around 85%. And of ABC transporters, 55% of them were drug efflux transporters, whereas 31% of total MFS transporters were drug efflux transporters. So this will be identified from the genomics. So now next we wanted to check what is the expression level of these drug transporters. If they are present in the genome, are they getting expressed also? So for that we did the transcriptomics by RNA-seq under two different conditions, in glucose as well as in Avicel. So we found that when glucose was given, majority of the transporters were not getting expressed, but in Avicel, a large amount of transporters were getting expressed, both of ABC and MFS. So you can see that towards FPKM levels, the expression levels of these transporters were higher towards the cellulose in both the conditions. So we found that so that means the expression of transporters was conditional. It was not uniform, it was conditional. Only under certain circumstances it was they were getting expressed. So carefully we chose the media accordingly. So we used three different kinds of media. One is SC, that is synthetic culture media, YNB, majorly YNB, and then we also used potato dextrose media. All fermentable sugars were there, and then we used a maltose based, a malt extract based agar. So we found, and then we so on these three media we added antibiotics at MIC level, and then we also thought that if we can, so if you can see here in the genomics part, that MFS was very important, because MFS transporters, 30% of them were of sugar transporters and other important factors which are important, which are essential for the survival of the fungi. So we never wanted to, we didn't try to block the expression or the function of MFS transporters. But if you can see here, ABC transporters, most of them were drug efflux. So we thought if we can add a blocker to the medium which can block the ABC transporter specifically, then it might affect the tolerance levels of the fungi. And for that we added chlorpromazine, which is a drug against the ABC transporters. Along with that, we also added small amount of non-ionic detergent. And then we found that both PDB and LMP, they were not showing any kind of growth in presence of these antibiotics. So now we know how to handle the fungi, and we can get clear results when we do the transformations or any genetic manipulations. So we can get the clear answer in yes or no. Because the growth will happen only when the gene will be present in this kind of optimized growth medium. And that's what we did. So we came to our third part, which is development of molecular toolbox. Here now we have tamed our fungi. So we tried optimizing the transformation protocols. And we found that agrobacterium mediated method was the most preferred method, and it was it was giving us very stable transformants. And when we use the optimized media for selecting the transformants, we found that almost all the transformants were positive. We never got any false positive results with the media, and all our transformants were positive. So first we expressed GFP into the fungi to see whether our transformation is really happening or not. And we could see as you can see here in the C figure. This A figure shows you how transformants looks like when they appear on the plate, on the selection plate. And so this is all dead material, and where you see the spores. So these are the positive colonies. And so these are the PCR confirmations for the cassettes that we had integrated. And these are the pictures so this is the untransformed, the normal wild type fungi, and this is the transformants, which shows GFP fluorescence. And we also confirmed this with the help of Southern hybridization. So once our transformation was done, our next target was to do the gene deletions in the fungi, because that was very important for our work, because we wanted to delete all kinds of negative regulators of cellulase expression. And for that, we optimized. So we found that agrobacterium was working very fine for us. So why not utilize this to delete the genes also? So for that, what we did, we took the tDNA and replaced it with the cassette having the homologous regions along with the hygromycin cassette. And that's how we were able to delete almost all the genes that we had selected for deletion. And so here you can see, so this is the plate of transformant. This is a transformant plate. This is the control plate, and this is our one of the transformant that is growing in the selection plate. And we confirmed the results with the PCR. And so we use different kinds of homologous, the length of the homologous ends and every time it was successful. And later on we realized that even as small as 500 base pair was good enough to delete the genes by this method. We also established CRISPR-Cas9 because we had to delete a lot of genes, and we had, and also at the same time, we need to overexpress many of the genes. So we also needed the CRISPR-Cas9 method. Again, for CRISPR-Cas9 method, we didn't use the plasmid-based method. Instead, we used in vitro method, where we provided the Cas9 along with the sgRNA, and we transformed this particular Cas9 and sgRNA into the genome by protoplast transformation. So first we made the protoplast of the fungi, and then we transformed Cas9 along with the sgRNA for the target site. And so here the results show you how we chose the 2 sgRNA. So we used 2 sgRNA to completely delete the gene. So one which is near to the start codon, a second sgRNA which is near to the stop codon. And by this method, we were able to delete almost all the candidate genes with ease, with a lot of ease. All right. So now we were done with the optimization of the tools, and we had made almost all the tools which were required for the genetic manipulations. So that next target was to rewire the signaling networks. So how do we increase the cellulase production in filamentous fungi? So there are 2 methods. So first method is to remove any negative regulation which hinders the expression of cellulases, or to overexpress all the transcriptional activators which increase the expression of cellulases. So we remove negative regulation by deleting the negative regulators, And we reinforce positive regulation by overexpression of the activators, transcriptional activators, as well as the genes, cellulase genes. So these are the methods how we increase the cellulase production in the filamentous fungi. So our first target was the major catabolite repressor, that is MIG1, which negatively regulates cellulase expression. It is a global negative regulator which hinders the expression of genes of secondary metabolism, almost all the genes of secondary metabolism. So this was not specific for cellulases. But removal of MIG1 becomes essential because if MIG1 is present, then cellulase expression of cellulase is minimal and under a lot of regulation, a tight regulation. So we identified the MIG1 from the genome, and we found that the MIG1 present in Penicillium funiculosum is a different allele because it has one stop codon at 134 amino acid position. So we named this allele as MIG1 134, but we found that the gene was functional, it was active, and it was able to regulate the expression of cellulases in the fungi. So our first target was to delete MIG1, and we deleted it. And we got the colonies in our very first attempt. And so then we wanted to see whether if MIG1 was really deleted, then it should show us two major classical tests that we do for checking the catabolite repression. So the first test is in the presence of allyl alcohol. So what is allyl alcohol? It's an allyl alcohol which is, if present in the medium, and if alcohol dehydrogenases, they are being expressed by the fungi, then alcohol dehydrogenases will convert allyl alcohol into a toxic compound, that is acrolein. But if alcohol dehydrogenases are not being expressed, then the fungi will survive in the presence of allyl alcohol. So as you can see, in the presence of glucose, when we give allyl alcohol, so the normal wild type strain should not express any alcohol dehydrogenases. So it will survive in the presence of allyl alcohol. But when we delete MIG1, the control over alcohol dehydrogenases is lifted and they are being expressed. And if they are being expressed, they will convert allyl alcohol into a toxic compound, which will not allow the growth. So you can see delta MIG1 didn't show any growth. So second test for checking the catabolite repression is if MIG1, so that is in the presence of 2-deoxyglucose. So what is 2-deoxyglucose? It's an analog for enzyme hexokinase, and if it is present, it will show the cell that since it is an analog of glucose, so cell falsely sense that glucose is present, and it will keep the cellulases and other secondary metabolic enzymes under check, and it will not allow the cell to produce that. So if deoxyglucose is present is given in the presence of cellulose, so 2-deoxyglucose is non-metabolizable. So if the fungi in the presence of 2-deoxyglucose, this fungus will be under the expression that glucose is present, so it will not produce any cellulase. So if cellulases are not produced, every cell is cellulose, so the fungus will not be able to grow in the presence of cellulose. But if the carbon repression is lifted, if MIG1 is deleted, then the fungus will keep producing cellulase even in the presence of 2-deoxyglucose, and it will show that carbon repression has been lifted. So these are the two experiments that we did to ensure that MIG1 has been deleted from the cell. So we found that when we deleted MIG1, it also helped the cell in other beneficial ways, that glucose utilization ability of the cell was increased, Sugar uptake was increased. As you can see, glucose utilization is also higher, as well as cellobiose utilization or uptake is also higher here by the MIG1 strain, by the MIG1 deletion strain. We also found that MIG1, when we deleted MIG1, the hyphal branching increased in the cell. The rate of hyphal branching increased. As you can see in NCIM, in the wild type strain, 72% of the hypha, they were found to be unbranched, whereas in MIG1, in the delta MIG1, almost 80% of the hypha, they were either bifurcated or trifurcated. So more hyphal branching was helping us because it was more because cellulase is only produced at the tip. It is not produced by the whole hypha. It is only produced at the tip. So more tips means more cellulase production. And that's what we found. So when we gave glucose and Avicel in different proportions to the cell, you can see the production of cellulase is much higher in MIG1 as compared to the wild type, specifically when the glucose levels are high in the medium. So we optimized the conditions and we found that the total protein production increased almost to the double levels when we deleted MIG1. And the total cellulase production also almost doubled with MIG1 deletion. So this was our first, so this became our parent strain. So MIG1 deletion strain is our parent strain. So rest of the modifications were done on the MIG1. So many modifications were done. So we did omics analysis of NCIM, or the wild type and the MIG1, and while comparing, we identified many transcriptional activators of cellulases. We checked or we overexpressed all of them, and we found the best one, and we expressed it. We also, using this analysis, we also identified all the signaling networks of cellulases, and we rewired the genes such that it enhances the cellulase production and secretion. While doing the omics analysis, we also found that which of the growth conditions are required, what all kinds of nutrients are required, and accordingly we optimized our media, which also increased. So we started with the 0.7 FPU. FPU is filter paper unit, that is measurement how we measure the cellulase in the secretome. So we started with 0.7 FPU. By a single deletion of MIG1, we reached up to 4 FPU per ml of the media. And then after multiple rearrangements and after multiple genetic mutations, we have now reached up to the level of 8 FPU per ml. So coming to the next and the last part of my talk, that is scale up and submerge. So once we were done with the genetic manipulation, so our next step was to produce enzymes at a large scale. So at lab scale, we optimized the production of cellulases up to 20 liter. And we used various methods. We optimized the pH, we optimized the best carbon source, that is best cellulose, which can help us produce a lot of enzymes. We optimized the best temperature. We also optimized other nitrogen sources, which were helping us in producing. And that's how we optimized at fermenter level. And here you can see this, we repeated the experiment at least 5 times with these conditions, with the optimized condition, and they were giving us the almost same results almost every single time. So these are the productivity levels of cellulase at 20 liter bioreactor. So once we were done at 20 liter, we tested at various other levels, at industrial levels. We checked at 200 liters level, 500 liters, 5,000 liters level, and then we also went for 20,000 liters levels. And at all the levels, we got almost the same productivity as we were getting in the lab. So once we were done with the optimization at fermenter level, we also, so I'll show you how the performance of our enzyme. So we used two different kinds of pre-treated. One is nitric acid pre-treatment and other is nitric acid and ammonia pre-treatment. So in nitric acid treatment, where most of the lignin is present. So even in presence of lignin, we can see that most of the cellulase was converted into sugars. So the lower graphs are of glucose and the upper graphs are of 5 carbon sugars that are xylose. So you can see even as we increase the enzyme, the hydrolyzing ability also increased, giving us higher amounts of fermentable sugars. And in both the conditions, so here only cellulose is present, so ammonia also removes the lignin. So here both only cellulose is present, and here cellulose plus lignin is present. And in both the conditions, we got almost all the activities. So so ammonia removes the xylose, removes the xylose. So this is pure cellulose here. And then we checked. So we also compared our enzyme with the market leaders, which is CTEC3 by Novozymes, and we found that at almost all the levels, enzyme levels, our productivity was much higher than the CTEC3 productivity. So our enzyme is a better alternative to CTEC3 for their use in second generation biorefineries, specifically when we use biomass of wheat straw or rice straw. So this is the total yield that we get. So 90% of the cellulose is converted into sugar when we use 5% of the enzyme with 15% biomass load. And and when with 3%, we were getting almost 80. So this was something which was industrially favorable at 3%. So this is our team. So here Dr. Shams Yashtani, who is the coordinator of the project at DBT ICGP Center for Advanced Bioenergy Research, and the rest of us. So I will thank you all for listening to me, and I'm open for questions if you have any.
Hello, Anmol.
Yes, sir.
Yeah, it was a very, very wonderful talk. We really enjoyed it very much.
Thank you, sir.
We liked it. And you have done a lot of fermentation work also.
Yes, sir.
Using a 20-liter fermenter, right?
Yes, sir. Yes, sir.
So can you please tell us a little more about the fermentation part?
Okay. So for fungi, we use aerobic fermentation. So here the supply of air is necessary, and we don't. So fungal fermentation, the cycle for fungal fermentation lies between 5 to 10 days. Because we get maximum productivity between 48 to 96 hours. So that's a time zone, and we have to optimize it according to the strain that we have. And so since enzyme is secreted out, all we have to do is to collect the media and centrifuge it, and you have your enzyme in hand. So we try to keep check on the pH, specifically
Yes.
pH. If the pH increases, the productivity it lowers. So we have to maintain the pH around 5 at fermenter levels.
And
So
what is the medium you use for the fungi?
medium?
Yes.
So It's a cellulosic medium, sir. It's not a clear solution. So
Okay.
it's a cellulose, and we also add some bit of hemicellulose, and we add a lot of protein in the form of maybe soya protein. That's the feed we use. So
Okay.
it's not a clear solution. It's not fermentable sugars, just like we do it in E. coli. So fungal fermentation is a little different in that
Right.
way.
Right. And
So
what is the
viscosity is a
temperature?
major problem.
Yes.
And breaking of hypha is another major problem for fungal fermentation. So we cannot increase RPM beyond a certain level.
Yes.
So these are the
So
key.
what is the temperature of your broth? Temperature.
Temperature is around 28 to 30. So fungi
Okay.
generally grows between 25 to 30.
All
So,
right.
so we optimize the temperature according to the strain.
Okay. And how do you maintain the fungi on PDA or on cellulose?
So we maintain the fungi on PD, potato dextrose, but for different performing different experiments, we keep changing our medium
I
according to
see.
the need.
What is the pH of PDA?
5.
5.8 or 5?
5. So 5.5 is the
Okay.
optimum one,
Yes.
but something around 5 works for us.
So 5 is very good. It is a bit, what to say,
Acidic.
yeah. Because in MTCC and Gene Bank, I used to grow the fungi and preserve on 5.8. It was
Oh.
recommended 5.8. But you are doing very good. 5 is a good pH.
For our fungi, so the strain that I was working on, it was able to grow even at 4.
Okay.
So it was acidophilic kind of.
Okay.
So 5 worked best for us.
Yes, very good. And temperature, you said how much temperature?
Around 28 to 30.
Okay, okay. So it is not a thermophilic fungi, it is a mesophilic.
No. Yes, sir.
Mesophilic.
GG.
Very good. And when you ferment it, is there a lot of frothing or there is no frothing?
D. There is frothing. Specifically in the first 48 hours, if we don't, we have to keep an eye on the frothing.
Okay. And does the enzyme also go to the froth sometimes?
So till 48 hours, enzymes doesn't start. So enzyme generally gets fungi start secreting enzyme after 48 hours. So frothing goes by till that time. So the fungal cycles is something like that fungi first grows and makes a lot of hypha, and then that hypha secretes cellulase.
Very good. You know why I'm telling is I was working on a lipopeptide, which is a surfactant, and most of the surfactant went to the foam. So we had to collect the foam separately, and this is known as foam fractionation.
Okay.
We collected the foam in a beaker, then we did acetone treatment, 3 volumes of chilled acetone, and we got all the surfactant in the foam, not in the broth. This is a very interesting thing. And this process is called as foam fractionation.
Okay.
Very, very good talk you have given, and we have learned so many new things from you. And any questions by anybody? Anybody wants to ask question? Okay, if there is no question, then I request Chetna to give a good word of thanks.
A marvelous evening to one and all. This is Chetna. Firstly, I would like to apologize for the delay as I couldn't join on time, but I'll definitely go through the YouTube recording. And thank you so much, ma'am, that you joined us today. There's always an end to every event, such as this auspicious event, and we are really glad to Honorable Dr. Anmol Randhawa, who joined us today. A heartfelt thanks to you, ma'am, and I missed more the half of the session, but I'll definitely watch it. And thank you so much, ma'am, that you take out time from your busy schedules and took your time for this amazing session. We are really looking ahead for such great sessions. Thank you so much, ma'am. Your presence has added an extra spark to this amazing interview session and made it even more special. खूबसूरत से एक पल का किस्सा बन जाता है, जाने कब कौन जिंदगी का हिस्सा बन जाता है। कुछ बहुत खास लोग मिलते हैं जिंदगी में जिनसे कभी ना बिछड़ने वाला रिश्ता बन जाता है। कौन किस पर मेहरबान होता है यहाँ, आप आए हैं ये आपकी मेहरबानी है। Thank you so much, ma'am, for taking out your time and graced us with your words. Thank you so much, ma'am. Finally, I would like to thank Honorable Dr. Arvind Deshmukh, who is the pillar of this Microbiologist Society India, Chandigarh. Thank you so much, sir, that you provided us this great platform for the panel discussion and for these amazing sessions. Now, I would like to thank Dr. Swarnjeet Singh, who is the president of Microbiologist Society India, Chandigarh, and I would like to thank you, sir, for organizing such great sessions. You are one of the top 2% global scientists of the world, and we are really lucky to have such amazing people with us. Thank you so much, sir. You have been awarded, lots and lots of award, and your main ambition is to reach to the remotest corners of this world and spread the knowledge of science everywhere. There is no limit of ambitions, and that they're not restricted to anyone. One can do all the best possible one is anxious to do, but every ambition must be fulfilled with a great will and sincere efforts for the fulfillment of it. A dream doesn't become reality through magic. It takes sweat, determination, and hard work. And it has purely been endorsed by all those greatest scientists that is available today with us on this amazing session. At last but not the least, I would like to thank everybody who joined us today and make this session a great success. At last, I want to end with these lines. खोल दे पंख मेरे कहता है परिंदा, खोल दे पंख मेरे कहता है परिंदा, अभी और उड़ान बाकी है, जमीन नहीं है मंजिल मेरी, जमीन नहीं है मंजिल मेरी, अभी पूरा आसमान बाकी है। Thank you so much.
Thank you, Chetna. You gave a wonderful word of thanks. Thank you, Anmol, for the nice lecture.
Thank you, sir. Thank you so much.
Thank you so much. Bye.